Good morning, all. Thanks for joining. Let us start with my topic presentation. So, the topic I choose is comprehensive analysis on the P53 genes of mammalians cell cycle regulations. So, let's start. So, the gene of interest chosen here is P53, which is tumor suppressed gene that is essential for controlling the cell cycle and halting the development of cancer cells. Here P donates protein and P53 show the weight of that genes. Example 53 kDa. You can see here. The expression vector I choose is CMV mammalian promoter. It is the potent and prevalent promoter and it is capable of promoting powerful transactions of the inserted genes. The aim of this project is clearly states in the topic that I wish to express the P53 genes in mammalian cells in order to look into any possible effects on cell cycles. So now here you can see the functions of P53 genes. Since we have selected P53 genes for insertions in the cell cycles, it is important to describe the main function of this protein. First of all, it's regulates the cell cycles, it helps in DNA repair, and it also promote apoptosis. Basically, it's prevent nucleoplastic transformation either by cell cycle regulations or apoptosis. Here you can see a figure that show a normal cell cycle which is consist of M phase, G1 phase, S phase, and G2 phase. Where G1 is a gap 1 and G2 is a gap 2. G2M and G1S are two important checkpoints, and in response to the DNA damage, P53 is induced in G1 phase of normal cell cycles. So now you can see my slide 4. To make it clear, let's have a look at what happens when there is a DNA damage. Any form of DNA damage is triggered the expression of P53 genes, which means to say that there is increased P53 levels. This increased P53 level is one of that prevents cells from entering S phase of the cell cycle. That means there is arrest of the cell cycles and G1 phase. It's low time. It's allow time for the DNA arrest of the cell cycles at G1 phase. So it's also allow time for DNA repair to take place. So the question arises that how DNA repair. Here P53 also induces more DNA repair genes which helps in DNA repair. Now two things can happen here. One, the DNA is repaired completely. Two, the DNA is so damaged that it seems to not be repaired. But when the DNA is repaired, there is no need of P53, so it's degrade and the normal cell cycle continue. While on the other hand, when DNA is not repaired, then P53 is sad. Either of these two things. One, it will cause permanent rest, or it activates the pro-apoptosis genes, and then it causes apoptosis. So from this whole cycle, we understand that whenever there is a damage to DNA, the P53 comes to it rescue. So let's move to next slide. So here you can see the impact of P53 on mammalian cell cycle regulations. So now we see the impact of P53 on the mammalian cell cycle regulations. P53 is considered to be a key regulator of the cell cycle. In response to the DNA damage, P53 can induce cell cycle arrest, apoptosis, and sense in mammalian cells. Control, sorry, in mammalian cells, and high production of P53 protein can be obtained by putting the P53 genes under the control of the CMV promoter. Increased P53 expression may result in greater responses to DNA damage and better preservations of genomic integrity through more effective cell regulations. So let's move to next slide. So in a nutshell, p53 genes insertions into CMV mammalian promoter causes highly constitutive expression of this protein, which show a potential impact towards cell cycle regulations, apoptosis, and senescence in response to DNA damage. So now you can see my summary here also. So you can read it in a nutshell. P53 genes insertions into a CMV mammalian promoter, causing highly constitutive expression of this protein. That's it. Thank you for joining. Bye bye.